Hello everyone. Welcome to Biology Crash Course for Bioinformatics. So basically, if you are just like me, who is not actually a biology person, but I'm from a non-biology domain, and if you want to explore the world of bioinformatics, then this course is actually made for you. So let me share a bit of my own experience. So I started working with bioinformatics when I was a master's student in my country, but it wasn't really hardcore bioinformatics. But later on, when I came into PhD in Singapore, I really started working with bioinformatics. So from master's to PhD, my understanding of bioinformatics and the biology of bioinformatics has grown dramatically. I can say that. But the issue is, I really wished that this kind of course was available before, because I will actually show you in the next slide the issue with the current courses and the current contents that are there. So basically, there's a lot of dependency in the biology parts. So if you're reading some paper and you are getting some bioinformatics or biology terms, in order to understand those, you need to understand a lot of other things, which if you try to find those in YouTube or in ChatGPT, you will need even more information. Let me give an example on this. So for example, my first work in masters, I did a research on this promoter. So there is something called promoter in our DNA. So I was trying to identify the promoter regions in the DNA. Now I was very good in machine learning even during my masters, because my undergrad thesis was on machine learning. So I was well equipped with that stuff and I was brought in as a machine learning guy. But when I read a few papers, I couldn't really understand the biology part or the biological implications because of this. So I didn't understand what promoter is. So I searched the internet and I found that in order to understand promoter, I need to understand what 5' end means, what gene means, and what gene expression means. I didn't know any of that. Okay, so now I started with 5' end. So when I tried to understand 5' end, I saw that I had to have some idea about the double strand structure of DNA, which I didn't know. Furthermore, when I tried to go into gene, I saw that I had to know about exons, allele, and mRNA in order to have a complete understanding of the gene in the human. And also, when I came into gene expression, which is obviously one step further from gene, I needed to know RNA-seq, FPKM, protein synthesis. None of those I knew. So you can see that quickly things are branching out. I will just show you one more step. So I saw that in order for me to understand mRNA, I needed to know the transcription process of RNA. In order to understand RNA-seq, I needed to know about PCR, complementary DNA. In order to understand FPKM, I need to understand DNA fragment. And for protein synthesis process, I need to have an understanding of codon and amino acid. So I can actually grow the tree much like broader, but I think you already have the idea. This is the major problem of biology content in the internet. So in this course, what I'm going to do is I'm going to arrange everything in such a way that you are not going to find this dependency. I mean, you are not going to find any struggle with this dependency because everything is in order. So if you just follow part 1, part 2, part 3, part 4, you are going to see that everything nicely actually goes into the flow and you are understanding everything while you are going forward. Now, I want to motivate you a little bit more. I mean, I'm going to show basically three paragraphs to you. These three paragraphs are very small portion of the research papers in bioinformatics or computational biology. I mean, the method is computational, algorithmic, but a lot of things are actually biology specific. For example, in this paragraph, you can see things like epigenetic modification, DNA, methyl group, post-replication modification, DNA strand, many other things, as you can see. Let me show you another paragraph. So here you can see alternative targeted cfDNA, mutations, variant allele frequency, cDNA level, gene panel, mutation clonality, copy number, clonal hematopoiesis, many other things. So, I mean, obvious that you wouldn't understand these things, right? Because you are not from biology background. These are biology things. But even in computational papers, even in algorithm specific papers of bioinformatics, you are going to see these words coming up all the time. Final example. So you can see there are things like non-invasive, shallow whole genome sequencing, copy number alteration, etc., GC content, these things. So again, I mean, the first research paper which I read after coming into PhD was on cell-free DNA, which is basically not a fundamental part of the bioinformatics biology, but it is basically a niche topic. But the papers were from Nature journals, and I mean, I would take 7 days just to read a 10-page paper. So basically, I was reading one page at a time in one day, and I would try to search all the, jargon words of bioinformatics and biology. I wouldn't understand half of it. I wouldn't understand half of it because of the dependency that I just showed you right here. So it's not easy, right? It's not easy. So that is why this course is here. So I will now talk about the course content, what exactly I'm going to cover from part 1 to part 8. So I'm just going to give you a brief overview just to give you an idea about what you are going to get in this course. First of all, I will talk about human DNA basics, where I talk about DNA location, chromosome double stranded DNA, etc. Then I will go on to talk about DNA sequencing and sequence alignment, basically the process of reading the DNA itself, the coverage, whole genome sequencing, targeted sequencing, these kind of things, and PCR, of course. Then I'll talk about copy number alteration and GC bias. So these are extremely important if you want to work with disease and different kind of cancer or other things. Then there's epigenetics. So if you work on human development or any type of development or any type of like niche topics, epigenetics and methylation will come in your way. So this will cover that part. Next, this is, I think, the most important lecture of the series, how RNA is transcribed from DNA and how RNA is translated to protein. So the central dogma theory. So this is perhaps the most prominent lecture in the lecture series. Definitely check that out. But again, I would recommend you to start from part 1, then gradually go down part 2, part 3, part 4, because then you are going to build the knowledge base that is required to actually go to the next parts. Then I will talk about gene expression. Now, here there are a lot of things like RNA-seq, ATAC-seq, etc., and how you measure gene expression. It's extremely cool lecture. Then we have the protein structure. So if you heard about DeepMind and AlphaFold, they are working on these topics, and this is a very exciting topic because now I think machine learning intelligence has already beaten the experimental procedures, which are very costly, so which is very cool. Then we have a DNA mutation. So mutation, I think we all heard about this before. This is not anything new, but it is actually new because we have heard that some chemical toxin is entering our body or some radiation, then human is becoming alien or things like that. No, a mutation is not like that. I will talk about everything regarding mutation, like all kinds of mutation, why it happens, the biology, etc, so that you actually understand DNA mutation papers and don't have to think about everything like a cinema or a Hollywood movie. Okay, so that is all that I will cover in this tutorial series. So I hope that you will bear with me and will enjoy the series. So if you enjoy the series, definitely feel free to like and subscribe to my channel, and I will try to upload more contents like this. So thank you everyone. Thanks very much.